Methods for monitoring and studying plankton distribution and abundance. If you're curious about how scientists track and study the tiny but vital world of plankton, you're in the right place. Let's explore the various methods used to monitor and understand these microscopic marine creatures. To start, plankton are collected using several techniques. One of the simplest and most traditional methods is the plankton tow. This involves towing a fine-meshed with cone-shaped net through the water to concentrate plankton in a sample bottle. This net can be towed horizontally to sample surface water or vertically to sample the entire water column. Another method is vertical sampling, which uses a CTD, conductivity temperature depth recorder, attached to a hose. This setup allows scientists to collect water samples at different depths, creating a detailed picture of the water column and the plankton within it. The hose is sunk to the bottom and raised, stopping every 2 meters to sample 10 gallons of water, which is then analyzed for both environmental data and plankton content. Once the samples are collected, they need to be analyzed. One common method for identifying and counting phytoplankton involves using an inverted light microscope, specifically the Uter-Mole method. This method requires counting phytoplankton from preserved water samples using different magnifications for different size classes of phytoplankton. For example, picoplankton, 0.2 to 2 micrometers, are counted at 1000x magnification, while microplankton, 20 micrometers, are counted at 100 to 250x magnification. A newer and more advanced method is the targeted volume correlative light and electron microscopy, VCLEM. This technique involves freezing samples at high pressures and then using a combination of light and electron microscopy. Researchers first use confocal microscopy to identify fluorescing cells and then use volume electron microscopy to reveal the 3D structure of the organisms. This method provides detailed images of the ultrastructure of plankton, including their protective coverings, mitochondria, chloroplasts, and other cellular components. For quantifying plankton biomass, several approaches are used. Phytoplankton biomass can be measured as biovolume, carbon content, or estimated using chlorophyll A, which is present in all phytoplankton organisms. The Phytoplankton Color Index, PCI, is another method used, especially with data from the Continuous Plankton Recorder, CPR, which estimates the green color of the plankton community. Molecular methods, such as DNA barcoding, are also employed to identify plankton species. These methods are often used in conjunction with morphological analysis to ensure accurate identification. This combined approach helps in identifying cryptic species and provides a more comprehensive understanding of plankton diversity. Monitoring plankton distribution and abundance over time is crucial for detecting changes in the marine ecosystem. Time series analysis is used to identify anomalies in plankton communities, which can indicate broader changes in the marine system. This involves collecting data at regular intervals over a minimum of 5 years and preferably 10 years to capture the temporal variability of plankton. In summary, monitoring and studying plankton involves a range of techniques, from traditional net tows and vertical sampling to advanced microscopy and molecular methods. Each method provides unique insights into the distribution, abundance, and diversity of these vital marine organisms.